Hi, my name is Dan Zuckerman from Health and Science University, and I want to introduce you to the weighted ensemble approach, which is a very general and powerful approach for simulating a wide variety of systems. So, start the PowerPoint here. This video is for people with experience in some kind of simulation method. Most of the lecture will be framed in terms of molecular dynamics simulations. However, really any type of stochastic simulation could be amenable to the method. And in particular, if you're running simulations where you can't see the phenomenon that you expect to see because they're too rare or too slow. So this is something that's very common in molecular dynamics simulations of complex biomolecules. Simple examples would include protein folding or binding. If we start simulations of systems hoping to see some of these phenomena, they really just don't happen at all using traditional simulation. And there won't be much technical knowledge needed for this lecture, just a minimum of equations, and I'm going to have references there for folks who want to read more and resources to link to. So let me focus for now on molecular dynamics simulation. So this is just a trace of the RMSD versus time. So the RMSD is the deviation from a reference structure. And this just shows the different states that a system visits over time. And this is a record-setting, actually, molecular dynamics simulation. It's a millisecond long. And what you see in this graph is that new phenomena are constantly emerging, even in this very small system where we don't expect too much to happen. We see even after hundreds of microseconds that new phenomena are emerging. For example, these different states here shown in black, these states shown in purple. So these are new things that emerge after quite a long time in the simulation. And for reference here, a more typical simulation would be in the, microsecond, single or a few microsecond timescales. So this is a rather extraordinary simulation, and it's very limited. And maybe in a more familiar context, if you just look at the timescales involved in molecular dynamics, most of the interesting processes and the most important for biology happen beyond the range that's really accessible via molecular dynamics. So we can frame this in a slightly more abstract sense. Basically, the larger the system you want to study, then the only way you can really sample it well is by coarse graining your system. If you want to have a very detailed description, atomistic or quantum, then you can only study a very small system. And the goal of a method like molecular weighted ensemble is to try to push out this boundary where we can do good sampling. And this type of picture applies for other fields, not only sort of systems biology, but really material science or any other field that you care to think of. So here's a key slide illustrating the basic idea of weighted ensemble simulation. And this is essentially a revision of a figure that was in the Huber and Kim 1996 paper. And although I won't be discussing history today, the basic idea of weighted ensembles actually developed much earlier and dates to researchers at Los Alamos National Laboratory, probably early 50s, late 40s, and the idea is credited to Johnny von Neumann. But here's the basic idea. You have your configuration space, and you cut it up into different parts. Your goal is, this black line is your energy landscape. to generate transitions from your initial state on the left all the way to the target state on the right. And this we presume to be a process that would be too slow by conventional simulation. So in weighted ensemble, what you do is you set a target number of trajectories in each of these bins that we've set up. So here there are 3 bins, and we set a target number of 2. So what you do, and actually instead of starting a single simulation, you start 2 simulations and you assign each of them a weight of 1/2. So the weights are noted down here. And you run these for a little while. And if you've designed your bins well, there's a good chance that one of those simulations will get to the next bin. So at that point, you basically check which bins are occupied and whether they're occupied with the target number of trajectories. In this case, neither of these has 2 trajectories in it, so you make an additional copy of each one. And these are identical copies, okay? And then, because we don't want to shift around probability, we simply take the probability of 1/2 and divide it into the 2 child trajectories. And then we go ahead and run each of these. And because they're stochastic, they don't go quite to the same place. And one thing that may happen, and of course we expect this from physics, right, is that systems will tend to go downhill in energy, and you may find yourself with more than 2 trajectories in one bin. In that case, there's a process by which you can prune away one of these trajectories and assign its probability to one or more of the other trajectories, and that's shown here. And in the second bin, however, you have only 1 trajectory again, and you make another copy of it. Okay, and now each of those shares the weight of the parent, which was a 1/4, and so they each get the weight of an 1/8, and then so on you proceed. So you can see that it's pretty obvious from looking at these weights that you can see exponentially rare occurrences. So that's the essence of the weighted ensemble idea. You can say it's a way of spreading your resources over your confirmation space. It's a way of seeing rare events that you couldn't otherwise see. So another view of the weighted ensemble algorithm is shown here in kind of a top view of a 2-dimensional system. And weighted ensemble is not restricted to 1-dimensional systems or 1-dimensional coordinates. You can use pretty much any coordinates you want. But here we say the same idea. Now we're using 3 trajectories as the target number. So imagine we start at this black dot, we use 3 trajectories, each has a weight of 1/3, and then the blue is the second iteration, and the weights change correspondingly as you do that. And then, in the same as the example we just saw, you can get a ratcheting effect and reach out to these much rarer bins, although with quite small probability, which makes sense. So here's another way of thinking about the big picture here. If you do a traditional simulation, what you'll do almost by definition is you're going to spend more of your computational resources in regions that are more probable, right? That's just the way simulations work. And so the region, however, you may be more interested in these tail regions of the distribution of possible outcomes. And that's where weighted ensemble really focuses resources. And it does it by construction, and it does it in a parallel way. So what I want to talk to you about today is shown here in this outline. Some of the strengths of weighted ensemble. I just mentioned the first one, the ability to see rare events. We'll go over these other ones. And then there are limitations, as with any method. And I want to talk about those. Some aspects may seem like a limitation in art, and others are a little more subtle. And then I'll talk about a few applications of the approach just to give you a taste of what it can do. So this slide has a technical name, and I apologize for that a little bit, but the point of this slide is that weighted ensemble is an unbiased method, and it's unbiased in the sense of this Fokker-Planck or Smoluchowski description. If you haven't heard of that, don't worry about it. It's basically, it is rather the analog of the diffusion equation for more complex systems. So you know that in simple diffusion, you have this equation that governs the probability distribution as a function of time. If your probability distribution starts as a delta function, it's going to spread as a Gaussian over time, right? So if you did weighted ensemble for a simple diffusion process, you would exactly recapitulate that spreading Gaussian. But likewise, if you have a system where there's a force, where f of x is a force, so the negative gradient of the potential, then it obeys a slightly more complicated equation where there's an additional term depending on the force, and this governs how the system, the distribution of the probability in this system will evolve in time, and weighted ensemble exactly obeys that. So that sort of thing is described in this paper. We also give a kind of resampling perspective on weighted ensemble and how it works, but the key point is it's not approximate; it's really an exact method. And this is just a little schematic showing how, if you have this gray double well potential, and then you start a distribution on the left, so a distribution you can think of as multiple trajectories, they'll sort of spread out over time, but a tiny, tiny fraction of them will reach over to the right. And this is exactly what weighted ensemble is going to give you, as we saw these exponentially small weights that occur. So wanted to show you some validation of this approach just to prove to you that it really works. And this was some work I did long ago. Basically took a protein, calmodulin, that underwent a conformational transition and modeled it with a very simple coarse grain model where only the alpha carbons of the protein are represented. And the advantage of that is that we could see these conformational transitions back and forth spontaneously. So here we didn't need any fancy algorithm to do this. It just went back and forth spontaneously. And we could even focus in on any one of these transitions, as I'm showing you on the right here. And we could see basically the fine details of those transitions as well. And there's a time scale that I'm going to come back to called the event duration, or sometimes called the transition path time, that measures how long it takes to make this transition. So this is one of the things that we use to validate the method. And what I'm showing you here is with a comparison of the distribution of that event duration or transition path time as calculated both by the weighted ensemble method and by just straight ahead brute force simulations. And you see that they're in very good agreement. However, if you used an equal amount of simulation time, what you see is that the estimate of the distribution you get from ordinary simulation is extremely noisy. So that's when I'm comparing the equal simulation time for the two methods. So that's a nice validation, and it shows what we call efficiency. We also can validate the approach by examining mechanism. In this case, we've just examined a plane representing a couple of distances between particular alpha carbons in that coarse grain model. And we can map every trajectory into this plane or project it into this plane. And then we can calculate distributions that occur as these trajectories cross these different planes that you see represented here. And you see the agreement is right on between weighted ensemble and regular simulation. So that's just confirmation that it's an unbiased method. Now, it's also a highly parallel method, and it's been used with many cores and different systems. Here's just a scaling plot to show you it's got this perfectly linear scaling because, and the reason is that there's just intermittent communication. Essentially, you running the dynamic simulations, and then only occasionally are the simulations talking to one another. Now, I refer to this aspect or this property that I called efficiency. And another way of saying that is that there's super parallel performance. And let me explain what I mean by that. So in a ordinary simulation, you might run a single trajectory on a single processor. And that's shown schematically. Here's the slide. Now, in standard parallelization, right, you basically, let's say you use 4 different processors. The goal is to regenerate exactly this trajectory, but just using a quarter of the time, right? So in some sense, in terms of your overall computing, you hope to break even, right? That's when you have perfect parallel scaling. However, in the weighted ensemble method, what we and others have shown is that you can actually get the key observables of interest, like some of the ones I was showing you, and also rate constants, in much less overall computing. So this is what we mean by super parallel performance. So it's a very nice property of this algorithm. I promised I would discuss with you some of the limitations of the weighted ensemble method, and I want to do that now. So one of them is that when you set up your bins in the space, as I showed you before, you're typically just going to use a small subset of coordinates, right? So imagine you're folding a protein, maybe you use just the RMSD, or maybe you use, for some conformational transition, the distance between 2 atoms. Of course, there are many other coordinates in the system. And if there are other coordinates in your system that are uncorrelated, then weighted ensemble doesn't help you sample them. And if they're both uncorrelated and slow, that means you're not going to make good observations of the process you're trying to see, if indeed those other coordinates, sometimes called orthogonal coordinates, are important. Now, I'm going to come back to this notion because there are some subtleties about it, quite a few, and I'm just going to sketch out one interesting thing. Now, another aspect, yes, and I should mention that this limitation I'm talking about applies to many other methods in this same sort of class of algorithms. Likewise, correlations are a problem that will occur in many of these advanced algorithms. So if you think about it, when we discuss the algorithm, the trajectories get replicated, right? And then that creates the ratcheting effect. But until the point at which they're replicated, right, those trajectories are exactly the same. So many of the trajectories that you get are highly correlated. So these correlations are essentially mean that you don't have quite as much information as it seems that you have. And the flip side of it is that when you if you do multiple independent weighted ensemble simulations, as I would urge you to do, you could see a large variance, that is, large differences between the runs. And that's just part of the process. That's inevitable. And another thing worth mentioning is that not every observable can be sampled more efficiently, right? It's only the observables that are slow that your bins help you to sample better through this sort of ratcheting effect that we discussed. Now, some of the things that may seem like limitations actually aren't limitations. And the thing I'm mentioning here is that you don't need to have prior knowledge of all the slow coordinates. And I'm showing you here an example of two, think of them as two hydrophobic spheres immersed in water. Now, I'm showing here a single reaction or progress coordinate R, right? But as you can imagine, the coordinates of the water are also very important. And it's almost impossible to imagine figuring out how to make bins for those coordinates to say track the behavior of the system as this R coordinate increases or decreases. Another way of saying it is these waters have to do some very complicated motions as you pull apart these spheres, or as these spheres spontaneously come apart, I should say. So do you have to know all the coordinates for the water? And the answer is you don't, because those coordinates are correlated with this separation distance. In other words, these two spheres cannot move apart unless the waters do their magic dance that lets them rearrange in the right way. So in weighted ensemble, we don't put any artificial forces on our system. We only use spontaneous motions that occur. And so if the waters move apart spontaneously, if the spheres move apart spontaneously, it's because the waters have moved out of the way appropriately. So we do not need to know about those. So coming back to the first point, these coordinates are highly correlated with the separation, right? I don't know what those correlations are. They're very complicated, but I don't have to know what they are. Another thing is I sort of suggested that, you make your bins and then they're fixed for the whole time. Well, they don't have to be fixed for the whole time. They don't have to be uniform. And in fact, really the bins themselves aren't actually intrinsic to weighted ensemble, but that's a more advanced topic. So just to give you a feel for this, here I'm showing you a sketch of a two-dimensional toy model that we studied with weighted ensemble. And the red lines are energy contour lines. So the X and Y axes are just two, coordinates, X and Y. And these are two basins on the left and the right. And there's these two pathways that kind of connect. These are two saddle points here. So if we start, let's say, a fixed number of trajectories all the way here, one of the things we can do is called a Voronoi procedure, where we essentially are using the trajectories themselves to make new bin definitions. And that's shown here as time progresses, going down and then across and then down again. And so basically the bins can be changed adaptively in time to follow the trajectories. So this is just something that we showed quite a few years ago, and it's an idea that's been taken up by other groups with some success. Now, one observable I didn't mention so far was the rate constant, which can be estimated as the inverse of the mean first passage time. So in other words, how long does it take to get from A to B if I start the system in A? And there's a relationship between the probability flux entering state B from A and this rate constant, or the inverse mean first passage time. And we pointed out a long time ago that this could be used to estimate the rate in weighted ensemble. More recently, we've developed some additional analyses that are related to Markov state models, but they use history information, so we call them history-augmented Markov state models. And these can be used to analyze weighted ensemble trajectories and even to reweight them for restarting. So this is an approach that we've started to use, and this is something that will be incorporated into the software. Let me show you some sample data from this. This is a protein folding study that we did on a few different proteins, or really two proteins, but three different models total. So one is the NTL9 protein at a low friction condition. And what you're seeing here in the red lines are estimates of that probability entering the folded state just directly from weighted ensemble simulation. So we call it WE direct. But another thing we can do is take the data from those simulations and actually use them to construct one of those history augmented Markov state models. And then we get the estimate shown in the green. What you see, which is reassuring, is that the red lines approach this green over time. And so those results are consistent if you can sample enough. Now, this was done under low friction condition, and that's, we believe, why there's a discrepancy between the experimental value and the value we estimate from simulation. If you go to, oh yes, one quick comment, that the uncertainty that you see in the range in these red lines is actually developed by using multiple independent weighted ensemble runs. And that's something I always recommend that you do, just to get a sense of how variable your runs are and to know how many of them you need to do. Now, if we go to a more water-like regime of viscosity or friction, then we see that, in fact, we're more in correspondence with the experimental value. And again, the red and green seem to be consistent. Now, in a more challenging system, this is protein G, we see something a little bit unusual. And this is, again, this is really the result of this high variability of weighted ensemble runs for challenging systems. And while it looks like we've reached a nice steady value, in fact, there's something quite artificial here. This is sort of making like a sharp right turn. And in fact, we're actually a bit undersampled from these direct simulations. And we can see that by putting this data through that HAMSM analysis, and we can get a much better estimate of the folding time from this. So that's something you can read more about it in our JAX paper. There's some other papers on this. Now, the weighted ensemble has been implemented in a few packages. The one that we use is called WESTPA. It's something that's developed primarily by my close collaborator, Lillian Chong. And there's a link here to the website with all the information about it, and a new version is coming out soon. Now, I've mentioned to you that weighted ensemble is not just for molecular simulations, and here's sort of a universe of weighted ensemble simulations that have been done in the biological space. And you see that some of them are actually just signaling networks, some are spatial models, virus capsid assembly, and then maybe the more traditional molecular dynamics protein studies. And not all of them are done by myself or my collaborators. Some of them are done by other groups. The ones noted in blue were done with the WESTPA software. And just to give you a sense for that, this is an example, really just a schematic of a signaling network model that was studied using weighted ensemble. And these models can get to be very complicated because there's a combinatorial number of species and reactions that occur. And for this particular network, one of the things that we looked at was basically the occupation of certain species that were difficult to access using standard simulation, which in this case wouldn't be molecular dynamics, but is actually done by a different approach called the Gillespie algorithm or stochastic simulation. And what you can see is that with weighted ensemble, we can see these extremely rare events that you couldn't see with standard simulation. So standard simulation sort of peters out here with let's say you did 1,000 different trajectories. So if we use that exact same amount of computing, we can see many orders of magnitude rarer events using the weighted ensemble approach. And similarly, this is just a kind of a toy spatial model. It's a cube where molecules are released from a top surface and then they can bind at the bottom surface. And this was done using a Monte Carlo simulation. It had to be the MCell software. And what you see here is a very similar result. There's a limit on how much you can see with standard simulation that's shown in the red line, and then we can see orders of magnitude rare events. So here we're looking at the number of bound receptors on that surface, which by the way was used as the progress coordinate for these simulations. So that gives you a sense of some of the different systems. So to sum up, the weighted ensemble method it's unbiased, it can be super parallel, it's very powerful. It yields mechanisms, rate constants, and other information that in an unbiased way with the right analysis. It doesn't depend on the scale of your system, the resolution, or the application domain. But it can't solve every problem, right? There's some systems that are going to be too complicated even for weighted ensemble, so don't make the mistake of thinking that this can solve every problem that you have, not necessarily. I will say that the WESTPA software is nice to use. It's open source, and there's a very helpful user community that provides a lot of support. So thanks so much for listening, and I really wanted to thank my longtime collaborator, Lillian Chong, who's a professor of chemistry at the University of Pittsburgh, and as I mentioned, her group does most of the development for the software. I've also listed here a lot of folks who've worked with me over the years on this method, and of course, the progress that I cited wouldn't have been possible without them, and we're very grateful for funding from the National Institutes of Health. Thank you.